Hello everyone. Today in this video I am going to talk about molecular docking. So first I will discuss about introduction, then definition, then role of bioinformatics, then types of docking, then drug discovery process, then types of docking software, then application of docking, then achievement in docking, and at last conclusion and references. So look at the abbreviation CAD, Computer Aided Drug Design, QSR, Quantitative Structure Activity Relationship, SARS, Structure Activity Relationship, STS, High Throughput Screening. Now introductions. Drug discovery. Take years to decade for discovering a new drug and very costly. Effort to cut down the research timeline and cost by reducing wet lab experiment, use computer modeling. Now drug target, enzyme, inhibitors, receptor, agonist or antagonist, ion channel blockers, transporter inhibitors, and DNA blockers. What is CAD? Computational chemistry is the chemistry whose major goal are to create efficient mathematical approximation and computer programs that calculate the properties of future drug molecules and helping in the process of drug design and discovery. So why CAD? Drug discovery today are facing a serious challenge because of increased cost and enormous amount of time taken to discover a new drug, and also because of rigorous competition among different pharmaceuticals companies. So molecular docking. What is docking? Docking attempt to find the best matching between two molecules. So whether the two molecules interact, if so, what is the orientation that maximize the interaction while minimizing the total energy of the complex. The goal to be able to search a database of molecular structures and retrieve all molecules that can interact with a Curie structure. So ligand or key, the complementary partner molecules which bind to the receptor ligands are most often small molecules, also other biopolymer. Receptor or lock, the receiving molecules most commonly a protein or other biopolymers. Docking, computational simulation of candidate ligand binding to a receptor. So why is docking important? It is the key to rational drug design. The result of docking can be used to find inhibitors for specific target proteins and thus to design new drug. It is gaining important as the number of protein whose structure is known increases. So why we do docking? Drug discovery costs are high. Drug interact with their receptor in a highly specific and complementary manner, and core of target based structure based drug design for lead generation and optimizations. So this is the traditional drug design. Now you can see the modern drug discovery process. So drug target, usually protein, binding of ligand to the target, that is the docking. So rational drug design, benefits, save time and dollar. So structure based drug design, you can see ligand database, target protein, molecular docking, ligand docked into protein active site, pharmacokinetics and pharmacodynamic optimization. These are the application of bioinformatics. Now types of docking, rigid docking and flexible docking. Now type docking, protein docking, both molecules are rigid. Interactions produces no change in conformation, similar to lock and key model. So protein ligand docking, ligand is flexible but receptor protein is rigid. Interactions produce conformational change in ligand. So protein binding site ligand. Some real docking, thermolysine protein with one of its known inhibitors. So available docking software like Dock, Flex, and these all are the available software. Now, application of molecular docking in the modern drug discovery: determine the lowest energy structure for the receptor ligand complex, search database and rank heat for lead generations, calculate the differential binding of a ligand to two different macromolecular receptor, study the geometry of a particular complex, propose modification of a lead molecules to optimize potency, other properties, de novo design for ligand generations, and library design. Application of molecular docking in modern drug discovery: case study like identification of inhibitor for simulation inhibitor of anticoagulation and anti-inflammatory activities of SNACs, venom, and phospholipase. Now, growing evidence of achievements. In conclusions, molecular docking gives the promising contribution to identification and optimization of ligand in modern drug discovery. The combination of the chemical information of natural product with docking-based virtual screening will play an important role in drug discovery in the post-genomic era as more and more new potential targets emerge from the functional genomic studies. Docking-based virtual screening lead to much higher hit rate than traditional screening method HTC. Now, future direction. So, computational medicinal methods can contribute its unique role in achieving the task of examining the interaction of all existing natural products with all possible targets and establishing their rational use. So friends, these are the references. I hope this video will be helpful to you. Thank you.